Okay, next let us discuss about regulation of enzyme activity, that is nothing but cooperativity and allosterism. Okay, let us discuss about this. Okay, here a significant portion of enzymes function such that their properties can be studied using the Michaelis-Menten equation. Okay, significant portion of enzymes function such that their properties can be studied using the Michaelis-Menten equation. However, a particular class of enzymes exhibit kinetic properties that cannot be studied using the Michaelis-Menten equation. Okay. However, particular class of enzymes exhibit kinetic properties. Okay. That cannot be studied using the Michaelis-Menten equation. The rate equation of these unique enzymes is characterized by an S-shaped, that is nothing but sigmoidal curve, which is different from the majority of enzymes whose rate equation exhibits hyperbolic curves. Okay, whose rate equation exhibits hyperbolic curves. Next, allosteric regulation is the regulation of an enzyme, okay, or other protein by binding an effector molecule at the protein's allosteric site. Okay, allosteric regulation means nothing but it is the regulation of an enzyme. Okay, it regulates the enzyme or other protein by binding an effector molecule at protein's allosteric site. Okay, by binding an effector molecule at where means at the protein's allosteric site, that is nothing but a site other than the protein's active site. Okay, next coming to the point, effectors that enhance the protein's activity are referred to as allosteric activators. Okay, effectors which are enhanced by the protein's activity are referred to as allosteric activators, whereas those that decrease the protein's activity are called allosteric inhibitors, which decreases the or reduces the protein's activity are called allosteric inhibitors. Okay, here the term allosteric refers to that fact that the regulatory site of an allosteric protein is physically distinct from its active site. Okay, here the term allosteric refers to the fact that the regulatory site of an allosteric protein is physically distinct from its active site. Okay, here allosteric regulation are a natural example of control loops such as feedback from downstream products or feed forward from upstream substrates. Okay, allosteric regulation అనేది a natural example of control loops. Okay, these are natural examples of control loops such as feedback from downstream products or feed forward from upstream substrates. Okay, here long range allosteric is especially important in cell signaling. లాంగ్ రేంజ్ అల్లోస్టెరీ అనేది ఎస్పెషల్లీ దేనికి ఇంపార్టెంట్ అంటే ఎందులో అంటే సెల్ సిగ్నలింగ్లో ఈ లాంగ్ రేంజ్ అల్లోస్టెరీ అనేది ప్లేస్ ఎ మేజర్ రోల్. నెక్స్ట్ కమింగ్ టు అల్లోస్టెరిక్ modulation, which is nothing but cooperativity. Okay, here cooperativity means it is a phenomenon displayed by enzymes. Okay, the phenomenon which are displayed by enzymes or receptors that have multiple binding sites. Okay, ఇప్పుడు ఒక ఎంజైమ్ తీసుకుందామండి, దానికి మల్టిపుల్ బైండింగ్ సైట్స్ ఉన్నాయి. ఓకే ఓకే హియర్ displayed by enzymes or receptors which have multiple binding sites. మల్టిపుల్ ఎలాగంటే ఇప్పుడు యాక్టివ్ సైట్ ఉంది, అలోస్టెరిక్ సైట్ ఉంది, అదర్ దాన్ దోస్ మల్టిపుల్ బైండింగ్ సైట్స్ ఆ ఎంజైమ్ చుట్టూ ఉంటే బైండింగ్ సైట్స్కి సబ్స్ట్రేట్ వచ్చి అటాచ్ అయినప్పుడు అనేది సబ్స్ట్రేట్ వచ్చి ఎంజైమ్ బైండ్ అవుతుంది కదండి. ఇప్పుడు ఆ యాక్టివ్ సైట్ ఉంది, అలోస్టెరిక్ సైట్ ఉంది, అదర్ దాన్ దోస్ సైట్స్ ఇంకా మోర్ దాన్ 2 సైట్స్ ఉన్నాయి, దాన్ని అదే మల్టిపుల్ బైండింగ్ సైట్స్ అని ఇక్కడ ఎక్స్ప్లెయిన్ చేస్తున్నారండి. ఓకే, అక్కడ ఏమవుతుందంటే రిసెప్టార్స్ దట్ హ్యావ్ మల్టిపుల్ బైండింగ్ సైట్స్ వేర్ ద ఎఫినిటీ ఆఫ్ ద బైండింగ్ సైట్స్ ఫర్ ఏ లైగండ్ ఈజ్ ఇంక్రీజ్డ్. ఓకే, అప్పుడు ఏమవుతుంది ఎఫినిటీ ఆఫ్ ద బైండింగ్ సైట్స్ ఫర్ ఎ లైగాండ్ అనేది ఆ లైగాండ్ దగ్గర అనేది ఇంక్రీజ్ అవుతుంది అనమాట. పాజిటివ్ కోఆపరేటివ్ ఆర్ డిక్రీజ్డ్ నెగిటివ్ కోఆపరేటివ్. ఇంక్రీజ్డ్ అయితే పాజిటివ్ కోఆపరేటివ్, డిక్రీజ్డ్ అయితే నెగిటివ్ కోఆపరేటివిటీ అని అంటామండి. ఓకే, అపాన్ ద బైండింగ్ ఆఫ్ ఎ లైగాండ్ టు ఎ బైండింగ్ సైట్. ఓకే, we also see cooperativity in large chain molecules made of many identical or nearly identical subunits such as DNA, proteins and phospholipids. Okay. When such molecules undergo phase transition such as melting, unfolding or unbinding, this is referred to as subunit cooperativity. Okay. Here cooperativity is a phenomenon displayed by enzymes or receptors that have multiple binding sites where the affinity of the binding sites for a ligand is increased. If the affinity of the binding sites for a ligand is increased, then it is known as positive cooperativity. If the affinity of the binding sites for a ligand is decreased, means then it is known as negative cooperativity. Okay, upon the binding of a ligand to a binding site. Okay, we also see cooperativity in large chain molecules which are made up of many identical subunits such as DNA, proteins and phospholipids. When such molecules undergo phase transition such as melting, unfolding or unbinding, this is referred to as subunit cooperativity. Okay, next coming to the point, an example of an positive cooperativity is the binding of oxygen to hemoglobin అనేది positive cooperativity అండి. One oxygen molecule can bind to ferrous ion of a heme. Okay, one oxygen can bind in an oxygen molecule can bind to the ferrous ion of a heme molecule in each of the 4 chains of a hemoglobin molecule. Okay, ఒక ఆక్సిజన్ మాలిక్యూల్ అనేది bind to ferrous ion of a heme molecule in each of the 4 chains of hemoglobin molecule. Okay, deoxyhemoglobin has a relatively low affinity for oxygen. Okay, deoxyhemoglobin కి వచ్చేసినప్పటికీ it has a relatively low affinity for oxygen. But when one molecule binds to a single heme, the oxygen affinity increases, allowing the second molecule to bind more easily and the third and fourth even more easily. Okay, the oxygen affinity of 3-oxyhemoglobin is approximately 300 times greater than that of deoxyhemoglobin. Okay, excuse me. The oxygen affinity of 3-oxyhemoglobin is approximately 300 times greater than that of deoxyhemoglobin. Okay, this behavior leads the affinity curve of hemoglobin to be sigmoidal rather than hyperbolic as with the monomeric myoglobin. Okay, this behavior leads to the affinity curve of hemoglobin to be sigmoidal rather than hyperbolic as with the monomeric myoglobin. Okay, by the same process, the ability for hemoglobin to lose oxygen increases as fewer oxygen molecules are bound. Okay, by the same process, ability of hemoglobin to lose oxygen, which increases as fewer oxygen molecules are bound. Okay, it increases as fewer oxygen molecules are bound. Okay. Next, negative allosteric modulation, which is also known as allosteric inhibition, occurs when the binding of one ligand decreases the affinity for substrate at the other active sites. Okay, negative allosteric modulation అనేది also known as allosteric inhibition, which occurs when the binding of one ligand decreases the affinity for substrate at the other active sites. Okay, for example, when 2,3-bisphosphoglyceric acid binds to an enzyme and an allosteric site on hemoglobin, affinity for oxygen of all subunits decreases. ఇక్కడ నెగిటివ్ అలోస్టెరిక్ మాడ్యులేషన్కి వచ్చేసినప్పటికీ నెగిటివ్ అలోస్టెరిక్ మాడ్యులేషన్ అంటే ఇట్స్ నథింగ్ బట్ అలోస్టెరిక్ ఇన్హిబిషన్. ఇట్ అక్కర్స్ వెన్ ద బైండింగ్ ఆఫ్ వన్ ligand decreases the affinity for substrate at the other active sites. Okay, for example తీసుకుంటే అండి when 3,2,3 bisphosphoglyceric acid అనేది allosteric site కి bind అయిందనుకోండి హిమోగ్లోబిన్ మీద, affinity for oxygen of all subunits అనేవి decrease అయిపోతాయి. This is when a regulator is absent from the binding site. ఇది ఎప్పుడు జరుగుతుందంటే regulator is absent from the binding site. Okay, another instance in which negative allosteric modulation can be seen in between ATP and the enzyme phosphofructokinase within the negative feedback loop that regulates glycolysis. Okay, another instance ఏంటంటే negative allosteric modulation can be seen between ATP and the enzyme phosphofructokinase within negative feedback loop that which regulates the glycolysis. Okay, here phosphofructokinase is an enzyme that catalyzes the third step of glycolysis, the phosphorylation of fructose 6-phosphate into fructose 1,6-bisphosphate. Okay, here phosphofructokinase can be allosterically inhibited by high levels of ATP within the cell. Okay, phosphofructokinase can be allosterically inhibited by high levels of ATP within the cell. Next, when ATP levels are high, ATP will bind to an allosteric site on phosphofructokinase. Okay, ATP levels ఎక్కువ ఉంటే ATP will bind to an allosteric site on phosphofructokinase, causing a change in the enzyme three-dimensional shape. Okay, this change causes its affinity for substrate, that is nothing but for fructose 6-phosphate and ATP at the active site to decrease, and the enzyme is deemed inactive. Okay, this causes glycolysis to cease when ATP levels are high, thus conserving the body's glucose and maintaining balanced levels of cellular ATP. Okay, this causes glycolysis to cease and when ATP levels are high, thus conserving the body's glucose and maintaining balanced levels of cellular ATP. Okay, in this way ATP serves as a negative allosteric modulator for phosphofructokinase. ఈ process లో ATP serves as a negative allosteric modulator for phosphofructokinase, despite the fact that it is also a substrate of the enzyme. Okay, here sigmoidal kinetic profiles are the result of an enzymes that demonstrate positive cooperativity binding. Okay, here sigmoidal kinetic profiles are results of enzymes which demonstrates the positive cooperativity binding. Okay, cooperativity refers to the observation that binding of the substrate or ligand at one binding site affects the affinity of other sites for their substrates. Okay, here cooperativity refers to observation that binding of the substrate or ligand at one binding site affects the affinity of other sites for their substrates. Okay, next, for enzymatic reactions with multiple substrate binding sites, this increased affinity for the substrate causes a rapid and coordinated increase in the velocity of the reaction at higher substrate complex until V maximum is achieved. Okay, until V maximum is. See here, for enzymatic reactions with multiple substrate binding sites, this increased affinity for substrate causes a rapid and coordinated increase in the velocity of the reaction at higher substrate level until V maximum is achieved. Here plotting the V0 versus substrate for a cooperative enzyme, we observe the characteristic sigmoidal shape with low enzyme activity at low substrate concentration and a rapid and immediate increase in enzyme activity to Vmax as substrate increases. Okay, see here, for enzymatic reactions with multiple substrate binding sites, this increased affinity of the substrate, okay, this increased affinity of the substrate causes a rapid and coordinated increase in the velocity. Okay, here సబ్స్ట్రేట్ ఇంక్రీజ్ అవుతుంది, అప్పుడు ఈ సబ్స్ట్రేట్ ఇంక్రీజ్ అవటం వల్ల వెలాసిటీలో కూడా ర్యాపిడ్ ఇంక్రీజ్ అనేది మనం చూస్తాం. ఓకే, of the reaction at higher substrate and velocity, okay, with V maximum is achieved. Higher substrate until the V maximum is achieved, okay. Here plotting the V0 and V versus substrate. ఇక్కడ మన వెలాసిటీ ఇనిషియల్ వెలాసిటీను సబ్స్ట్రేట్ ఒక గ్రాఫికల్ రిప్రజెంటేషన్ ప్లాట్ చేసుకుంటాం మనం ఒక గ్రాఫ్ని for an enzyme cooperative enzyme. We observe the characteristic sigmoidal shape, okay. It's see here, this is the characteristic sigmoidal shape curve, okay. Sigmoidal curve which represents the presence of cooperativity, okay. With low enzyme activity. Enzyme activity అనేది ఇక్కడ తక్కువ ఉంటుంది at low substrate concentration and a rapid and immediate increase in the enzyme activity to maximum velocity as substrate increases. The phenomenon of cooperativity was initially observed in the oxygen hemoglobin interaction and with that functions in carrying oxygen in blood, okay. Positive cooperativity implies allosteric binding of the ligand at one site increases the enzyme affinity for another ligand at a site different from the other site, okay. Enzymes that demonstrate cooperativity are defined as allosteric. There are several types of allosteric interactions. An example, homotropic and heterotropic. Here homotropic means positive and heterotropic means negative. Positive and negative allosteric interactions like refer to the enzyme's binding affinity for other ligands at the other sites, okay. For other ligands at the other sites as a result of ligand binding at the initial binding site, okay. Here positive and negative allosteric interactions refers to, that means homotropic and heterotropic allosteric interactions refers to enzymes binding affinity for other ligands at other sites, okay. As a result of a ligand binding at the initial binding site, okay. When the ligands interacting are all the same compounds, the effect of the allosteric interaction is considered homotropic, okay. Ligands interacting are all the same compounds. Effect of the allosteric interaction is considered as homotropic when the ligands interacting are different, okay. Ligands interaction అనేది different గా ఉంటే effect of allosteric interaction అనేది heterotropic అంటాం. Ligands interaction అనేది same compounds అయితే అది homotropic, okay. It is also very important to remember that allosteric interactions tend to be driven by ATP hydrolysis, okay. Next, coming to the point, Hill equation, okay. Let us discuss about the Hill equation. That is nothing but degree of cooperativity which is determined by Hill equation for non-Michaelis-Menten kinetics, okay. For non-Michaelis-Menten kinetics, the Hill equation accounts for allosteric binding at sites other than the active site. Here n is the Hill coefficient, okay. Here n is the Hill coefficient. మన ఈక్వేషన్ తీసుకుంటే theta equals to L power n by kd plus L power n which that equals to L power n by kNa plus L power n, okay. ఈ ఈక్వేషన్ తీసుకుంటే ఇక్కడ theta అనేది ఫ్రాక్షన్ ఆఫ్ ligand binding sites filled, okay. L అనేది ligand concentration, kd అనేది apparent dissociation constant which is derived from the law of mass action. K వచ్చేసి K power n అనేది K is అనేది ligand concentration producing half occupation, okay. Ligand concentration which is producing half occupation, that means ligand concentration occupying half of the binding site, okay. That is also the microscopic dissociation constant K అనేది. n అనేది Hill coefficient మనకు తెలిసింది, that describes the cooperativity. See here, దీనిని బట్టి మనం ఒకసారి మళ్ళీ ఈక్వేషన్ చెప్పుకున్నది theta equals to L power n by kd plus L power n that equals to L power n by Ka power n plus L power n, okay. ఈ హిల్ ఈక్వేషన్ తీసుకొని taking the logarithm of both sides of the theta of the equation that leads to an alternative formulation of Hill equation, that is log of theta by 1 minus theta equals to n log L minus log kd. Okay, where when n is less than 1 there is negative cooperativity, when n is equals to 1 there is no cooperativity, when n is greater than 1 there is a positive cooperativity. Okay, next allosteric models, that is nothing but allosteric. Currently there are two models of illustrating cooperativity, the concerted model and the sequential model. Most allosteric effects can be explained by the concerted model. Okay, that is nothing but NWC model put forth by Monod, Wyman and Chaincux. Okay, allosteric models ఉన్నాయి కదండి, సీక్వెన్షియల్ మోడల్, కన్సర్టెడ్ మోడల్ అనేవి ఎవరు ఇంట్రడ్యూస్ చేశారంటే ఈ మోడల్ని Monod, Wyman and Chaincux అనే సైంటిస్ట్ ఇవి ఇంట్రడ్యూస్ చేశారు. బై ద సీక్వెన్షియల్ మోడల్కి వచ్చేసినప్పటికీ ఈ కన్సర్టెడ్ మోడల్ ఏమో Monod, Wyman and Chaincux అంటాను. సారీ, కన్సర్టెడ్ మోడల్ వచ్చేసి Monod, Wyman and Chaincux. Sequential model ఎవరు డిస్క్రైబ్ చేశారంటే Koshland, Nemeth and Filmer. Okay, both postulate that enzyme subunits exist in one of the two conformations, that is tensed or relaxed, and that relaxed subunits bind substrate more readily than those in the tense state. Okay, the two models differ most in their assumptions about subunit interaction and the pre-existence of both states. Okay, there are two primary models for illustrating cooperativity. Okay, two ఏంటంటే ఇక్కడ చూడండి, this is the allosteric model diagram చూసుకుంటే పాజిటివ్ అలోస్టెరిక్ ఎఫెక్టర్, పాజిటివ్ అలోస్టెరిక్ సైట్, అలోస్టెరిక్ యాక్టివేషన్ జరిగి ఏమవుతుందంటే కోఫాక్టర్ బైండింగ్ ఏరియా దగ్గర కోఫాక్టర్ అనేది వస్తుంది యాక్టివ్ సైట్ ప్రెసెన్స్లో. ఓకే, ఇది కెటలిటిక్ ఏరియా. నెగిటివ్ అలోస్టెరిక్ ఎఫెక్టర్, నెగిటివ్ అలోస్టెరిక్ సైట్ వచ్చేసినప్పటికీ అలోస్టెరిక్ ఇన్హిబిషన్ జరుగుతుంది. అప్పుడు అంటే ఏమవుతుందంటే బైండింగ్ ఏరియా అంటే సబ్స్ట్రేట్ వచ్చి బైండింగ్ ఏరియా దగ్గర అటాచ్ అవుతుంది. ఇది కెటలిటిక్ ఏరియా, ఇది బైండింగ్ ఏరియా, substrate. Okay, this is the overview of allosteric models, allosteric activation and allosteric inhibition. Next, concerted model గురించి తెలుసుకుందాం. The concerted model of allostery also referred to as symmetry model, that is nothing but దీన్ని సిమెట్రీ మోడల్ అంటాం, లేదంటే Monod, Wyman, Chaincux మోడల్ అని కూడా అంటాం. ఓకే, this postulates that enzyme subunits are connected in such a way that a conformational change in one subunit is necessarily conferred to all other subunits. Okay, here conformational change in one subunit is necessarily conferred to all other subunits. Thus all subunits must exist in the same conformation. Anni subunits ekkada exist avutayante same conformation lo exist avutayi. Okay, the model further holds that in the absence of any ligand, that is substrate or otherwise, the equilibrium favors one of the conformational states that are tense and relaxed. Okay, here the equilibrium can be shifted to the R or T state through the binding of one ligand to a site that is different from the active site. Okay, the equilibrium can be shifted to the R or T state through the binding of one ligand to a site that is different from the active site, nothing but allosteric site. ఓకే, నెక్స్ట్ సీక్వెన్షియల్ మోడల్కి వచ్చేసినప్పటికండి, సీక్వెన్షియల్ మోడల్ అనేది ఆఫ్ రెగ్యులేషన్ allosteric regulation holds that subunits are not connected in such a way that a conformational change in one induces a similar change in the others. Okay, here a conformational change in one induces a similar change in the others. Okay, thus all enzyme subunits do not necessitate the same conformation. That means moreover the sequential model dictates that molecules of substrate binding via an inducer fit protocol. Okay, inducer fit model, that means substrate binds to the enzyme other than the active site, which is nothing but allosteric site. Okay, here In general, when a subunit randomly collides with a molecule of substrate, active site in essence forms a glow around its substrate. Okay, it forms a glow around its substrate. While such an induced fit converts a subunit from the tensor state to relaxed state, it does not propagate the conformational change to adjacent subunits. Okay, ikkada it does not propagate the conformational change to adjacent subunits when such induced fit converts a subunit from the tensor state to relaxed state. Okay, instead substrate binding at one subunit only slightly alters the structure of other subunits so that their binding sites are more receptive to a substrate. Okay, instead, substrate binding at one subunit only slightly alters the structure of other subunits. Okay, so that their binding sites are more receptive to the substrate. Okay, to summarize this sequential model, subunits need not exist in the same conformation. Molecules of substrate bind via induced fit model. Okay, conformational changes are not propagated to all subunits. Okay, this is the overview of cooperativity and allosterism. Okay, thank you.